Let's start by talking about prokaryotes. How are prokaryotic genes regulated? DNA-binding proteins in prokaryotes regulate genes by controlling transcription. DNA-binding proteins are proteins that will, as you might be able to guess by their name, they bind onto the DNA. They stick to the DNA. And then they regulate genes by controlling transcription. To conserve energy and resources, prokaryotes regulate their activities using only those genes necessary for the cell to function. By regulating gene expression, bacteria can respond to changes in their environment, for example, the presence or absence of nutrients. How does an organism know when to turn a gene on or off? One of the keys to gene transcription in bacteria is the organization of genes into operon. An operon is a group of genes that are regulated together. The genes in an operon usually have related functions. The E. coli, shown in the picture, has operons within its DNA. There are 4,288 genes that code for proteins in E. coli, including a cluster of 3 genes that are regulated together for the metabolism of lactose. Lactose is a sugar. To digest lactose, E. coli needs certain enzymes. The 3 genes that code for such enzymes in E. coli are called the lac operon. Why must E. coli be able to switch the lac genes on and off? Lactose is a compound made of 2 simple sugars, galactose and glucose. To use lactose for food, the bacterium must transport lactose across its cell membrane and then break the bond between glucose and galactose. These tasks are performed by proteins coded for by the genes of the lac operon. This means that if the bacterium grows in a medium where lactose is the only food source, it must transcribe these genes and produce these proteins. If grown on another food source, such as glucose, it would have no need for these proteins. The bacterium is able to detect the presence of lactose and therefore when the products of these genes are needed. When lactose is not present, the lac genes are turned off by regulatory proteins that bind to DNA and block transcription.